Thanks, Naveen, and thanks everybody for being here. So I guess I want to do a couple things. The first is I want to thank the DVOC and Nate Rice, who couldn't be here tonight, also wants to thank the DVOC because, you know, over the years, we've gotten lots of support from you for our undergraduate student research program. And in fact, today, I received the latest check from, from the treasurer to, to help support that program going forward. And it's great. This really gives us a lot of flexibility to bring in students. And instead of having to have students volunteer, which is really kind of looked down upon in today's day and age, we can pay students to do work in the ornithology department. And that might be work. Oh yeah, I can try to get here in the screen. That might be work like, helping Nate in the collections and, curatorial oriented stuff, but it's also getting this experience doing research projects. And I think there's nothing more motivating than knowing that you're going to actually get paid to do the stuff that you enjoy and that you love. And it really helps to kind of lower the barrier for students to be able to come in and do this kind of work. So we appreciate it. I know the students appreciate it. And I hope that we can continue to do this kind of stuff in the future. So what I wanted to do was, just give you a little bit of a quick overview of, what we're doing in the department a little bit. And then I want to talk about a little bit about what other people are doing in the department with research, and then I'll introduce the two students and what they're doing. So I'm just going to touch on what a bunch of us are doing, not really myself, although I'll sort of touch on the themes of that. So it'll be a little brief overview of the work that we're doing, what our current postdocs, graduate students, and undergrads are doing, and then I'll introduce Abigail and Matthew, who are going to talk about their research. So this is just a slide that shows you, the kinds of stuff that we do with specimens in the collection. And I'm not going to sit and go through this list, but there's all kinds of great stuff that can come out of specimens in museum collections. And this is, real world conservation. We collect conservation genetic data. It can be, the pictures that are in your field guides. Those are based on specimens in collections. All kinds of great things that we can do with the material that we have here at the Academy. And we're involved in a lot of this. The collection here has over 200,000 specimens in it, around 87,000 species, 8,700 species. So a pretty good proportion of the species of birds in the world. So it's a really useful and important collection. And it's not just used by us, it's used by lots of people. We often think about specimens and we think, these specimens, they don't have a lot of data. Well, we do all kinds of things. And these are just sort of a smattering of the different kinds of things that get collected with regard to any specimen that gets prepared for our collection. So we don't necessarily do all these things, but we do many of them for any given specimen. So modern specimens are filled with all kinds of things that can allow us to answer all kinds of questions, including the kinds of questions that Abigail and Matthew are going to be talking about. They're going to be talking about things that deal with parasites, that are ancillary materials that many museums historically did not collect off of their specimens, and many still don't, but we do. We actively collect that kind of material off of our collections. And one of the cool things about working in collections like this is we're sort of, we're doing modern science, but we are also doing this really traditional kind of work, this traditional museum-based ornithology, but we're doing genomics at the same time, so we are really sort of spanning these two worlds, and it's everything from like really basic exploration, figuring out what creatures are on our planet, to understanding what the base pairs of the DNA are in their genomes. So kind of a cool, diverse array of things. And these pictures here are just showing, some of the kinds of things that we do. So that's Nate in the middle in the field on Ilha Marajó, which is an island in the Amazon, at the mouth of the Amazon River, where we did a field expedition a few years ago. And then there's Mel Carricker in the left-hand corner, who was a collector for the collection here and many other collections in the United States. And he's one of the reasons that we have one of the best collections of Andean birds in the world. He also happened to be a louse biologist. So he described more lice species than anybody alive at the time. There's now one person that has beat him in terms of number of species described. But what's cool is that, we're at this institution where Curator worked and where Curator put a lot of his specimens, not most of his louse specimens though. Those are mostly in the Smithsonian, but we're doing that kind of work now too. So it's kind of a neat connection, like a cool historical connection. And this is just sort of a smattering of the kinds of categories of things that we work on. We work on avian evolutionary biology in the ornithology department. We do a lot of work on coevolution of birds and their parasites. And then we also do biodiversity surveys of birds and their parasites. And a lot of this involves genetic work and genomic work and building genealogies, but it involves other kinds of descriptive work too. And, you know, we're going to be talking about parasites when, you know, Abigail and Matthew will be talking about parasites. And this is one of my favorite statistics, that 30 to 70% of life on Earth is parasitic. This also happens to be a segment of life that we don't know a lot about. And there's a lot of interesting questions that we can ask using parasites. So this is just a diverse array of some of the parasites that are found on birds and inside of them. Genomics has been a really big thing in our lab. You know, when I started out, I was working my rear end off to get a few hundred base pairs of DNA data from one fragment of mitochondrial DNA that's matrilinear transmitted from organism to organism. And that could tell us some stories, but not like the stories that we can tell with whole genomes. And the two students that are going to talk to you today are sequencing whole genomes from organisms. You know, so they're collecting, you know, millions of base pairs, you know, of DNA data, which is crazy. And it's a really different world, even the way that we analyze the data. So that's a pretty cool situation. And this picture, you're going to sort of see some of this again, is from a project that we're working on toucans that that involves trying to use genomic data to look at the coloration of birds. So we can use it to study all kinds of cool characteristics. We still do these traditional museum expeditions because we need material to study, right? So since I've been here, we've been on 3 expeditions to Amazonia and Brazil, 1 to Mexico to the Transvolcanic Range. We've had 2 expeditions go to Nicaragua. We've done field work here in Pennsylvania. And then we also do a lot of collections building by salvaging specimens. And we've done that all over the place, Pennsylvania, New Jersey. Barb has helped us a lot bringing specimens from Cape May that are in their freezer, and those are being put into the collection here. We collect parasites off of those. We collect tissue samples and they get prepared for our collection. And then we do a lot of salvage by trading things with other museums. So that's been a great way to build our collections and add to our research programs. And then this is just some of the places that we've gone to in Amazonia and Brazil. So it's like real exploration of unknown places. And then we come back into our lab and we explore unknown things about the genomes of these organisms. Pretty cool stuff. So let me just touch on what a few people are doing in our lab. So one of the cool things about a vibrant lab like ours is that we have a whole slew of different kinds of trainees. You know, there are researchers, we have postdocs, and the first 2 people that I want to introduce to you are postdocs. One of them, I think a lot of you already know, but a lot of you probably do not know Luke Musher. He's actually working remotely now, but is here in Philly sometimes, usually once a week. And Luke is a postdoc, did his PhD at the American Museum, and he is a specialist on Amazonian birds. And one of the sort of broad questions he's asking in a lot of his research is, why are there so many birds in places like Amazonia? And he studied this from the perspective of studying particular groups and the population genetics of these things. So one of the birds he's studying is this white-shouldered antbird. And he's actually looking at how river movement affects diversification. So kind of a unique area in evolutionary biology. Am I, is it this? Sorry. Oh, how do I get rid of that? Do that. Thanks. That's great. Okay. And I think a lot of you know Therese Katnack. I don't know if Therese is still a member of the club, but Therese definitely came to meetings, has spoken to the DVOC about some of their work on birds of prey. And this is just a big evolutionary tree of raptors that Therese published actually with Matt Halley. Matt Halley was one of the co-authors on this. And Therese and I have a big NSF grant to study accipiters and all their close relatives. So to study their evolutionary history, how all these different body sizes have evolved in these raptors over time. And Therese's work, the nice thing about having scientists like Therese and Luke around is that these people are experts that are training our students how to do things that I can't do, right? So it's like having 3 of me with different areas of expertise. And it's a great way to sort of, you know, everybody's training each other. So it's a great part of a vibrant department. Very recently, and some of you may bump into Samantha because Samantha's a crazy birder, as is Luke and Therese. Samantha is a new collections assistant that we just hired in January. And Samantha is actually working on a cool project where we are rehandling every specimen. Our goal is to ultimately rehandle every specimen in the ornithology collection and verify its data. We know that there are lots of specimens in the collection that were never database. So in a sense, if we search our database, we don't even know we have them. We might find them in the drawer, but they're not in that database. Of course, we find out when someone puts in an information request when we go to a drawer that there's something not database sometimes, but this is the best way to get at that. And also the best way to get all the data that's on those labels into the database that is not there. So when it was originally database, they did not capture every data field. So we're enriching the database, and Samantha is to thank for that work. And Samantha's working with a bunch of interns to get that going. Now, what's cool about Samantha is a big bird of prey lover, has worked with birds of prey, has worked with bird banding stations that ban birds of prey, and actually worked on a project studying hippobossid flies. So this picture here is a hippobossid fly with lice attached to its abdomen. So lice actually ride on these flies. So Samantha's an expert on these hippobossid flies. And that's one of the other cool things about parasites. It doesn't take you long to become one of the world's experts because there are not that many people that know a lot about a lot of these things. So Samantha truly does know about them and knows about their distributions and knows how to identify them. So that's pretty awesome. So someone that loves birds, loves working with specimens, and also is working with these cool insects that we hope to study more in our lab. Now I want to talk about a couple of my PhD students that are in the lab. This is Camila Cuabara. Camila is from Brazil, and I first met Camila when she was an undergrad in one of my colleagues' labs at University of São Paulo at the Museum of Zoology there. And she was studying ectoparasitic lice with them. And she's truly one of the world's experts on parasitic chewing lice. She's a taxonomist, knows the morphology of these things, but also through her PhD has learned how to process next generation whole genome sequencing data on these things. And Camila's working on a whole bunch of ecological and macroevolutionary studies aimed at understanding things like how these parasites co-evolve with their hosts in terms of body size. There's a hypothesis out there that the larger the bird body size, the larger the parasite body size. And in some groups that's true, in some it's not. She's actually testing that hypothesis with the parasites that she's studying. And this is actually the host of the parasites that she's studying in the center here. She's working on tinamous. We have a big NSF project that's about to wind down that was funded to study the phylogenetics, the evolutionary history of tinamous and their parasitic chewing lice. They happen to have incredibly diverse faunas of lice. Some of these things can have 19 species of lice found them. If you compare that to a robin has 4. So this is incredibly hyper diverse community of parasites, and Camila is studying them. And the other thing about Camila is she's going to stick around the ornithology department when she defends in the spring. She is now our new lab manager. So some of you may have met Janice Despato, who was my lab manager for years. She retired and we were able to fill her position, and Camila was a great fit for that position because she knows the molecular work and the parasites. So we're excited to have her stay on in the ornithology department. This is John Merwin. John is another PhD student, and John is working on this toucan system. And this is a system that I started working on for my PhD, but I never could have dreamt of the kinds of things that we can do to study these things. So there's mimicry in these Ramphastos toucans. Basically, if you look at that bird up at the top, Ramphastos swainsoni, that is thought to be a model for Ramphastos brevis, the choco toucan. And basically, these small-bodied croaking toucans are supposedly the mimics, are supposedly mimicking the large-bodied yelping toucans. And John is actually studying this in the context of plumage coloration. So there are Amazonian ones that have the same thing. And we're actually using these genomic sequences to try and understand what the genetic underpinnings are for these physical characteristics. So a pretty exciting project. And there are some other aspects to his PhD as well. This theme of studying color and genomics is sort of becoming a bigger thing in my lab. And Emily Griffith, somehow I forgot to get her name on this slide. Emily Griffith is also a PhD student in my lab. And I think Emily spoke to all of you about her project on owl UV coloration and fluorescence. So Emily is working on the evolution of dichromatism in dabbling ducks. So I think all of you are familiar with dichromatism in dabbling ducks. You have mallards that males and females are different. You have some ducks where like Chiloe widgeons in South America, where males and females are bright, they're monomorphic. And then you have some things where you have monomorphic dull, like black ducks. And she's trying to understand what are the genes tied to this kind of phenotypic trait. We know something about the evolution. We think that dichromatism was actually lost, that bright males, that we see the loss of that color over evolutionary time. But she's going to actually be reconstructing that on more modern phylogenies with modern data, and then also looking at the genetics of this in a variety of ways. So we can compare genomes and look for sort of hot spots of differentiation that can tell us that genes are associated with these characters. It's also a pretty cool project on a cool group of birds. Oh, I did have her name, but somehow it was not up right away. And then Calvin Keys is one of my undergrads. And unfortunately, Calvin couldn't come today. He has a Spanish class that he's taking at night. And he's also on his co-op working at Willistown Conservation Trust, which I know a lot of you have spent time at WCT at their reserves, probably bird watching. And Calvin started out as a STAR student. So we have a program at Drexel called Students Tackling Advanced Research. Basically, the university pays for students to come and work in a research lab. And Calvin started in our lab on that and worked on this project on these white-winged bicards. And white-winged bicards are kind of an interesting bird because if you look across the tropics, they vary in the amount of dark coloration. If you look at males, they have this geographic variation in plumage coloration. And one of the hypotheses out there is that this is what we call ecogeographical variation. Basically, there's a correlation between things like precipitation and temperature and the coloration. So the general hypothesis out there, the general rule is that the more humid the environment, the darker the plumage. So he's testing that by measuring specimens. We're looking at body size as well across the geographic distribution. And then we're taking special photos. We have a special set of, we actually have two cameras, one that has visible light, one that does visible and UV. The UV filter's been removed from that camera. And we can quantify that by using special color standards and a special UV standard. So Calvin has worked on that on this group of birds, and Matthew, who's sitting here and is going to talk to us, did that for a project that he started with one of my PhD students. So at some point, we'll get Calvin here to come and talk about his project. This is a new student in my lab, Kai Doran. He's an intern right now, but is going to be a STAR student this summer. And Kai kind of came from the interest of dinosaurs, and of course, birds are dinosaurs, and was interested in morphology. And he is working with Emily Griffith, the student that works on ducks, to study the evolution of spines on waterfowl. So a lot of waterfowl and many other birds have these crazy spines. So the one up at the top there is one from a screamer, which is sort of the sister group to all ducks, geese, and swans. So he's studying the evolution of that using an evolutionary tree, and is just starting that project this summer, is going to be doing CT scans of specimens to do that. Okay, now we're to our two students who are here. And I can't remember, Abigail, are you going to go first?
I can, yeah.
Or
I said I lost the rock, paper, scissors.
Oh, okay. Okay, so Matthew's going to go first, or?
I'm going to go first.
Oh, you're going to go first. Okay, Abigail's going to go first. I'm going to introduce both of you anyways, and then I'll introduce you before your talk as we get them up. But so Abigail has been in our lab for several years, started out as an undergrad, but she's what's called a BS-MS student, which means you can do 4 years of undergrad and you do a 5th year as a master's. And a lot of students will do that just with a course only kind of master's, but Abigail has chosen to do a master's thesis. And that's what she's going to talk to you about, her master's thesis, where she's getting close to defending. So she's working hard these days working on that. She's been working on, a set of parasites that we collected from, or that colleagues of ours collected from prairie grouse. And she'll tell you more about that. But Abigail's worked on lots of things in the lab, everything from specimen prep to basically reorganizing our entire egg collection. And she's been an integral part of our lab over the last bunch of years. And she's graduating and actually has a job. So we're very excited for her, but we're going to miss her too. So that's Abigail. And that was her working at the scope, sorting lice, probably from the grouse. And it was like right as the pandemic, it was at a point when we were probably still wearing masks around the museum. I think we took that shot without a mask because most of my shots had masks at that point. Okay, Matthew Soesanto is an undergrad, is currently a co-op in our department, and this is his second time doing a co-op in our department and has also been an intern during the school year. And Matthew started out working on this project where he was helping John Merwin to quantify coloration in these Ramphastos toucans. And that actually led us to a recent trip. We went to LSU, Matthew and John and I, to measure all the toucan specimens there for their coloration, because there are a lot of those that we're sequencing the genomes of in that collection. And this is just sort of a picture showing, how we're photographing these things, and then how these, these spots that we're analyzing for coloration sort of can be analyzed using, a couple of software packages. And, Calvin and Matthew and John have been sort of integral in getting that kind of stuff going in the ornithology department. And it's really cool stuff. It's going to be integrated with genomic data. Back in my day, all we said was upper tail coverts red, upper tail coverts yellow. We scored them. We didn't actually quantify them. So pretty cool that we can do that now. Okay, so that is all I have. So I'm going to pull up their presentations. Actually, oops. Hard here. There we go. Oops. Well, did not mean to unshare, but we'll reshare in a second. Okay, Abigail. Let's see. So Abigail is going to talk to us about her project on whether agriculture affects infection and genomic variation in prairie grouse lice. Abigail. Is that okay?
I'm gonna get my notes.
Oh.
So hang tight for a second. No. Do you have speaker view set up? No, it's okay.
Yeah, it would not do speaker view, unfortunately.
Just in case.
Yeah.
I'm gonna
Grab your laptop.
But
Are you gonna do a good setup?
so while I get this ready, thank you for having me. I really appreciate it. I've been a student in Jason's lab for I think over 3 years now, which is kind of great. And yeah, this is part of my ongoing research for my master's thesis, which I'm defending in about a month. So that's intense, but
Is that gonna work okay, Abigail?
this will be,
I can also, if you want me to advance for you or something, so you can.
it's okay.
Okay.
I'll make it work.
Okay.
If I block them with the laptop.
No.
Okay, that's fine.
I was just trying to figure out what's comfortable for you.
We'll make it work. So broadly,
What you have to, have share.
Oh, shoot, I didn't share, sorry. That's my fault. Yeah, it's not like it's mirroring to the two screens. That's, yeah, that's the, okay. Okay, so now we should be good.
so my project is sort of situated in like a growing concern about observations of biodiversity declining worldwide, especially for grassland species. And as we know, grasslands are home to many important and charismatic bird species like greater sage grouse that we all care about, and lots of other animals and plants as well. And this decline of grassland biodiversity coincides with agricultural intensification. And this began in the mid-1900s and is marked by things like increased use of pesticides, herbicides, creating these homogeneous monocultural landscapes and habitat disruption. So this map shows the decline of grassland bird species around the world. All of the colored spots you can see on the map represent about 400 different research papers, all of which report on grassland bird species being in decline specifically as a result of agriculture. And the main threats that agriculture presents to wildlife is that it breaks up native prairie or grassland habitat and this habitat is of course replaced with crops. So if we examine this problem a bit more locally, we observe declines of many species, which are all of the red areas on the map. And these are mostly happening in areas that we have a lot of heavy agricultural production. So because this large-scale shift in land use has occurred in relatively recent history, it's possible to examine its effects on a finer scale and not look at how it affects species, but how it sort of works within species, and ask how wildlife is being affected by agriculture at population level. And for my project, of course, I'm not really looking specifically at birds, but I'm looking at lice that are found on 2 species of bird hosts, greater prairie chicken and sharp-tailed grouse. These birds have many similarities, including their diet. They have lecking breeding behavior that they're famous for. They're closely related, and they also have hybrid offspring. And we know that agriculture probably has negatively affected them, like greater prairie chickens range, for example, is so much smaller than their historic range used to be. And again, like what I'm more interested in looking at is not how agriculture has affected the birds, but how it's affected the things that live on them. These prairie grouse share the same kinds of lice. So for 2 of the 3 genera or groups of lice on each bird, Amorcidia and Goniocodes, they share the same species of lice. And for the Louse genus Lepidopithecus, there's a different species on each bird. The one on greater prairie chicken is undescribed. The takeaway here, though, is that the grouse share lice from the same louse lineages. And I'll briefly go over a bit about what avian lice are. They're flightless insects that parasitize birds. They are called obligate permanent parasites, meaning that they complete their whole life cycle on their hosts. They spend their entire lives on their hosts. That's where they reproduce. They need for resources. And they're also considered to be very host specific. And what that means is that one species of louse often only infests one or a few species of birds. And while many lice do show strong host specificity, this louse that we found does not. Goniocodes gallinae is also called the fluff louse, and it parasitizes domestic chickens and is considered to be an agricultural pest. It's been found on other wild birds that are sort of related to chickens, but as far as I can tell, this is the first time it's being reported on greater prairie chicken and on sharp-tailed grouse. And the other interesting thing is that in my sampling, this louse greatly outnumbered the native host-specific lice that I was expecting to find. So maybe another impact of agriculture on parasite diversity that I hadn't really considered was that introduced parasites as a result of humans may be able to outcompete native specialist parasites. For other lice, though, there's this general expectation of them being strongly linked to specific hosts. And what's interesting about such close host-parasite relationships is that over time, birds and their lice can develop similar evolutionary histories and similar population structure. And if that's an unfamiliar term, you can basically think of it as subgroups within the larger population that are grouped together by genetic similarities. The idea being that they're sharing some sort of common ancestry. This figure is an example of Galapagos hawks and their population structure, as well as the population structure of their lice. So what we're looking at basically are 2 bar plots. One for, so like the one on top, each bar is representing an individual hawk, and the different colors represent different ancestries that show us the different subgroups of the hawk population. So if we look at like the bar in green for Espanola, the individuals for the most part share one common ancestry, which is why it's mostly a green bar. And if we look at the opposite end of the plot, where there's lots of colors, you can see that the individual hawks just have much more variation in their DNA. So they probably come from multiple different ancestries. And you can see that in the louse plot, below the hawk plot, that the lice are very closely tracking the variation of the subgroups or the population structure of their hosts. And now I'll just outline the setup of my study. Again, I have my 2 grouse hosts, 3 genera of lice, 2 types of host habitat, and I'm asking basically 3 questions. First is, does habitat type, or like the host habitat type, whether they're from cropland or prairie habitat, affect their probability of being infected by lice? Oh, two number ones. I'm also asking how are louse populations structured? And I swear on Google Slides, that was not an issue. And lastly, what variables are actually responsible for that structure? So I'm testing to see if distance between host individuals, the identity of the host species, and host habitat type have an effect. I sequenced genomes from 75 different lice, and I identified SNPs, which you can basically think of as sites on the genomes that have variation. And that variation can be used to show population structure. And from that variation, I used two different methods of visualizing it and tested the effects of the different variables, again, distance, host species, and habitat type. I tested whether agriculture increases the probability of hosts being infected using what's called a generalized linear model. This is just the raw data of the prevalence on hosts from different habitat types. And just sort of at a glance, it looks like there might be more lice on hosts from prairie habitat. So maybe that means that hosts from prairie have a greater probability of infection. And to actually test it and not just eyeball it, the results of the model showed that prairie hosts are more likely to be infected with the lice Amricidia and Lagopicus, but are less likely to be infected with Gonia codes, which is interesting because that's the agricultural parasite. For my second question, how are louse populations structured? This is a visualization of the population structure for Lagopicus. Again, there are bars showing each individual's genetic variation, and you can see that the plot is basically divided in half by two different colors. This means that there's two ancestral sources of variation, and it appears to be coming from the two different species, almost perfectly. So orange matches lice from Greater Prairie chicken or sharp-tailed grouse, and blue matches lice from Greater Prairie chicken. And this pattern of variation being split basically by host is consistent with all three kinds of lice, not just Lagopicus, but that's my example for now. So this is another way of visualizing genetic variation, also in Lagopicus. And again, host species appears to be having an effect. So on this plot, the red triangles are lice from grouse and blue circles are lice from the prairie chickens. And when we have individuals clustered together in a group, it means they're more similar, genetically similar. And because all of the red triangles are clustered on one side and all of the blue on the other, this is representing how genetic variation of the lice is separated by host species. And what's interesting is that while Amricidia also kind of had that split halfway through, here host species doesn't seem to be causing that pattern. And when I zoom in, you can see that lice from different host species are overlapping each other. So while that population structure does exist, because I did find it, host species doesn't seem to be the cause of it for this specific group. So if host species is causing population structure in Lagopicus, it's not clear what's causing it in Amricidia. And that kind of gets into my third question about what's actually causing it. Again, I tested host habitat, host species, and distance between hosts. And I also tested the combined effects of these variables to see if together they have a stronger effect. So this is what's called a commonality analysis plot. It sort of shows the effect that each variable has on population structure. And all you really need to pay attention to are the blue dots in the very center of the plot. And these are representing the effects of each variable. And because they're all basically in perfect alignment with 0, that means the analysis is telling us that they have no effect. And I got the same finding for the combined effects of the variables. However, this is kind of strange because we already saw that variation, in Lagopicus is almost perfectly predicted by host species. And I ended up finding out that this method isn't able to handle like perfect prediction, and it kind of just can't interpret that information. So this plot is giving me the wrong information, almost certainly. And the results are pretty much the same for Amricidia. None of the variables are very predictive of the structure that I found. So what I need to do now is try to find another approach and see what that tells me. So to wrap up with my conclusion so far, habitat type does affect the probability of infection. Again, I think it's really interesting that hosts from cropland are more likely to be infected by an agricultural pest from chickens. The louse populations were structured with 2 sources of genetic variation, with host species almost certainly playing some level of a role in this. But although it partially explains some of the structure, the method I'd used just wasn't appropriate for assessing the contributions of all of the variables, and I'll need to find another way of doing this. So that's all I have for now. Thank you for listening.
Or wait,
It's up to you.
I'll see if anybody has any questions. Anybody have any questions right now? Check the chat.
You mentioned distance, a slide or two back between host and host something, but you're talking about a statistical distance?
No, geographic distance.
Oh, okay.
Yeah, so if you're familiar with the idea that birds, for example, on islands can become more
Yeah.
sort of isolated, basically the same thing is happening with lice on individual birds. It's hard for them to disperse relative to other organisms. Yeah.
This might have been a question for Jason, came on a little while ago, but Perry asked, what makes parasites so important?
I have answers,
Yeah.
but I think, so, well, this is also kind of, I feel like this is getting into your talk. Okay, not to give up the ghost here, but the interesting thing about parasites being able to so closely track kind of like what's going on in the population genetics of their hosts is also that if they have faster generation times, they reproduce more quickly. So they're tightly linked to these hosts, but they're also accruing like generations and evolving faster than their hosts are. So you can kind of hopefully use that information to help you conserve species of importance. But that's what I feel like that was, I don't want to spoil too much, but that's my
Yeah,
answer.
and they're also, and that's one great reason. I mean, they're models for understanding the evolution of free living organisms too, because, like Abigail was saying, they live on hosts like free living organisms live on islands. They're, the hosts are kind of the geography of parasites. So they're really cool models for understanding, how organisms evolve. Like we can like we always talk about, like, we're trying to understand how birds evolve. And we need to understand something about geography or geology to understand that. That may be harder to reconstruct, but we can reconstruct the kind of geology or geography for a louse by reconstructing the evolutionary history of its hosts. So we can build a phylogeny, these genealogies or evolutionary trees of the hosts. And then we can compare that to the genealogy or evolutionary trees of the parasites. And, what the history, was of what that parasite lived on. So it's kind of cool, they're the habitat for these things. You can measure all kinds of things from that habitat. The interbarb width of the feathers that they insert between to when they're trying to hide from the host preening them, for example. So there's all this really cool evolutionary biology. There's cryptic coloration. So if you look at like black cockatoos and white cockatoos, the black cockatoos have black lice, the white cockatoos have white lice. This is repeated over lots of different groups of birds where you have different coloration patterns. The lice are often cryptic in color. So there's all this really neat evolutionary biology.
Sounds like pepper moths.
Yep, exactly. It's like the pepper moth thing. And there have been some cool experiments done. So should I introduce Matthew?
Yes. Oh, maybe one more question.
Okay.
Sure.
Victor asks, and Jason and Abigail say that the birds on the islands for the parasites, are they talking about the bird species or the individual birds?
Both.
Yeah, exactly. Both. It depends on the story. The neat thing is some of these parasitic lice can ride on hippobossid flies and have a way to move around. Some we think can walk on their own a bit and move around. There are duck lice that we think disperse between ducks by riding on little feather boats on duck ponds. And those are generalists. So we see a lot of variation out there in the louse world. Like some things are really specific, some things aren't. So, the same thing is true, like some of these are like an island. An island would be a whole species that they specialize on, but there's also a lot of population structure on individuals like Abigail stuff. So you can study these things at really macro evolutionary scales and really micro evolutionary scales and ecological scales, and it's all kind of cool stuff from my perspective.
And do you know if these host-specific relationships are true for non-birds, mammals, reptiles?
Yeah, some of the models the first model to study this was gophers and their lice. People, and gophers are not very social. They don't come into contact with each other, and people started studying co-speciation between the gophers and their lice. And that was really sort of like one of the foundational things that led to this kind of work. So I don't think, am I still sharing? No. Like unshares, I guess, when I click out of the. I missed something. Okay. Okay, so I'm going to introduce Matthew Soesanto, who's going to talk to us about whether feather louse genomes reflect the bottleneck events of host population. I'll let him tell you about the specific group he's studying himself.
Thanks, Jason. Hello, everyone. Welcome to my talk about my research project, which involves California condors, lice, genomes, DNA, all that cool stuff. But before I get into all of that, let me give you guys a little crash course on the lice I'm studying. So this is a California condor, for those who don't know. Largest current bird in North America, large wings, large feathers. And when you dig deep into those feathers, you'll see feather lice that parasitize the bird. Now, there's two types of lice species found on this bird. The first is Copocephalum californici. This is the species that we've known for a while, and we go way back. In the 1960s, we've known these lice to be present on this bird. And they're the ones that tend to be the smaller and more rounded bird. I think they call it generalists, so they're found on the general body of the bird. The other species is Falco-Ipurus. It's actually undescribed yet right now, so we don't have a name for it, but they're the larger, longer lice found on the wings. So what I have here is a timeline that tells the general story of the California condor and its parasite, the louse. We started in 1963, where Roger Price first described this Copocephalum californici. And, it's pretty cool news, finding a new species. But unfortunately, during this time, the California condor was already in great decline. And this was due to DDT, poaching, lead poisoning, habitat loss. And then it went so bad that in 1967, we declared this species as critically endangered. Not much happened. A decade and a half later, the population continued to decrease. And in that year, we had 22 individuals left in the wild. Number was so bad that we decided to start capturing these birds to regrow the population through a captive breeding program. The problem to that is that in the process of capturing and taking care of these birds, the caretakers would treat them with pesticide that killed off all the louse, all the lice on the bird, and presumably driving this California condor louse to extinction. The story doesn't end there, because in 2018, we actually rediscovered the lice again. And after that, we started to discover more lice on other California condors. And this is because we think basically the caretakers didn't do a good job in killing all of the lice on the birds. So some of them still continue to survive and live on throughout the whole years. And in addition to that, the breeding program that happened in the 1980s, it worked. So now the condor population continued to grow, and in 2020, we have more than 300 individuals back in the wild. So all these events are all part of a phenomenon we call a bottleneck effect, which is basically when a population undergoes great decline in size. And what's interesting about these effects is that they're actually detectable through studying the genomes of species. So let me explain a little bit on that. Imagine you have a jar full of marbles, and you have all sorts of marbles. You have white, red, and green marbles, a high diversity. And let's say you lose some. Let's say your friend puts their arm in a jar, grabs a handful of marbles, and you lose, coincidentally, all the white marbles. So now you're left with red and green ones. Now imagine even harder here. Let's say those marbles started reproducing and laid eggs and regrew back in population size. So you're back to the original number of marbles in the jar. The difference is that now you only have red and green marbles. And this kind of changing diversity happens in the wild. It happened with the California condor story, where you have this population of condors undergo a population change or decline and allow them to regrow back in population, and you have a change in diversity. But instead of colored marbles, in this case, it's the genome diversity in these condors. And through studying the genomes of these condors, we can detect bottleneck effects. So let's zoom out. What does this have to do with my project? Well, this first paper, published by Robinson et al., talks about genome diversity in the California condors and other related New World vultures. And their paper basically said that they detected multiple bottleneck effects present in the history of the condors history. So in that figure, you can see that the x-axis is time, the y-axis is population size, and the more right you go along the x-axis, the more into the past you go. So as you can see, those hills basically show great declines and regrowths in the condor's history. In addition to this paper, there's another paper that talks about the advocacy for parasite conservation. And they basically touch on what we talked about just now, about parasites being important tools to inform about host history. And that's basically because for parasites, they live so close to their hosts, basically on them or in them sometimes. And they have shorter generation times. So that means they have quicker evolutionary rates, so they can inform us more about the host with greater resolution. And sometimes there are cases where dealing with host genome is more difficult. Maybe the genome is bigger or more complicated and it requires more computational power, things that we don't have yet sometimes. So studying the parasite may be a good alternative. So combining these two papers, I have two main objectives. The first is to use the first paper analyses and to apply them to lice genomes. Where they studied birds, I'm studying lice genomes. And my second objective is to compare those two analyses to see if there's any correlation between the genomes of the lice and the host. And I'm basically trying to ultimately answer the question, can parasites inform us about the history of these hosts? So in addition to California condors, I'm also studying the lice found on related vultures, such as black vulture, turkey vulture, and greater yellow-headed vulture, sorry. And that's basically because I just can. I have the data set for it, but also I want to see if there's anything or similar results or supporting results compared to the California condor. And I've basically broken up my project into 3 steps. First is imaging, where I just take pictures of these lice. And that's because in the next step, I'm going to crush these specimens to extract DNA from them. So, imaging is always a good way to go back and ID these specimens. And it's not as easy as using my phone to take a picture. These guys are really small. So we actually have a camera setup that takes multiple pictures, that focuses on multiple parts of the lice, and it mashes them together to get this crisp, detailed photograph. After imaging is DNA extraction. It's a 2-day experiment where I extracts DNA from those specimens. And then once I get that, I send them to a company for them to sequence. They do their magic. It's whole genome sequencing, for those who might know. And what they give back to me is this huge file that contains all these DNA strands. And my next step, which I'm currently in right now, is to take all those little strands and construct a whole genome out of it. And once I do that, that's when I can start doing my analyses and tests. And this step is very new to me. I'm new to coding, new to all this analysis part, but I'm learning, and yeah, that's pretty much my project. So I want to thank basically the people in the ornithology lab who've held my hand every step of the way, answered all my questions, and thankful for that. I also want to be thankful to the institutions who actually sent us the Louse specimens, because they're the ones who are actually dealing with the birds, the lice. And thank you all for listening.
Any questions in the room? Just a couple
So
questions.
how would you compare the lice on the condors with the lice on the other cultures?
So there's already a paper that Robinson's paper already has results for analyses of the bird. And my plan is to use the same method of analyses with the same kind of tests and apply them to the lice genomes and to see if the lice show this kind of pattern or not in their history as well.
Couple questions on Zoom. I had a very similar question. Perry asked, Perry says that she's curious if the lice are injurious to the birds they infest.
All right, what's it?
If the lice are injurious to the birds.
Like if they injure the birds, do they cause problems to the birds?
To a degree, yeah. If you have a lot of parasites on you, that definitely affects your health. But I will say that there's a good amount of balance, of like, because these parasites also don't want to kill their host because they feed off of them, and if the host dies, then the parasite dies.
Yeah, there's a lot of like, basically, if an individual bird has a really high load, then they have a really negative effect. If it's just a few parasites, a few lice on it, it's not going to be as negative effect. And there have been experiments done on some groups of birds, like rock pigeons. People have studied those, and they know that rock pigeons and also swifts, when they have high loads, they have lower reproductive success. But most of these birds probably don't have a super high load. Like, we know that parasites have what we call a skewed distribution. Basically, very few individuals have a lot of parasites, and most individuals have a few or no parasites. Although these big birds probably tend to mostly all have some.
Are more gregarious species, do they tend to have a higher load?
Yes, that's the general correlation. And actually, Camila, my PhD student, is studying these kinds of questions in actually, we've done all these Amazonian collections, and this is actually material collected when I was at the Field Museum before I came here and since I've come here. So she's sort of looking across a whole swath of Amazonia and looking at, all the species that we sample and trying to understand these kinds of comparative questions. Like, is it nesting behavior of the host? Is it, what explains the intensity of infection on these things, or the prevalence of infection, or the probability of infection?
Is it possible to get lice off of museum specimens, from 100, 200 years ago?
It
Do
depends
analyses
on,
that way,
yeah.
and get DNA out of them?
So I think it would depend on the preparers, how they skin the bird, because back in the day, I don't think they checked for a lice, right?
Right.
But now,
Curator may have, Curator did, but
But they could still be attached. On the other hand, you'd have to consider cross-contamination in the
yeah,
tray.
so there are lots of lice on museum specimens. Like, if you look at the toucans that were in my photos,
Yeah.
you look at them all closely, they all have nits, the eggs of lice on their feathers. But you can also find lice on the birds. The problem is that when you do that, there are a couple issues. One, we have not been successful at getting DNA out of those because they're so small. We could probably get DNA, but not much. And you need quite a bit of DNA to sequence a whole genome. And we're already dealing with the edges of that with a teeny louse as it is. Normally, we like to keep a voucher. We like to slide mount them. But when we're doing whole genome sequencing, we can't do that. We can only do that if we want to look at a few genes. Then we can get enough DNA to look at a few genes. So that's why we're doing these high quality photos. If you sort of know what you're looking for, you can go to museum drawers and you can get material. And actually, Matthew and Camille were trying to do that for king vultures and a few other things, because they're trying to figure out what this Falco-Lipurus is. It's a new species that's on California condors. It's undescribed. So they want to know what's on other things to compare, and we don't have specimens of some of those things. So they looked at our specimens in the collection. I think I know one.
we shaped around 10 huge birds. And
Yeah.
we would think that we would find at least one, but yeah.
Sometimes you do. So I did this for a project on trumpeters and lice from trumpeters in Amazonia, because we found a new genus on trumpeters, and it's very rare. We only had about 5 specimens, so we wanted more for our study. So we went to birds. This is when I was at the Field Museum. We went to the collection and ruffled those. And we actually got tons of lice off of those. And we found the louse we were looking for, but we also found seabird lice and all kinds of lice that we know are not from those hosts. So
Cross-contamination.
yeah, you don't know how the host has been handled, basically. And when we sample things, we do know how they're handled. We bag them separately. We're very careful about that.
Greg has a couple of questions on the chat, related to Barry's question earlier. Is there any known benefit to the host from the presence of ectoparasites, such as those being discussed?
Yes,
To
to the host.
To the host.
Well, I guess the very definition of parasite is to take, well, parasite benefits the host that it benefits.
Abigail may have.
So I don't know if there are examples of this specifically in birds, but there are some instances where native parasites can provide a host defense against like a parasite that got introduced. So I think it's happened in trees, and I think there's something also with moose and elk happening where their range, basically the northernmost species can't get very far south without being decimated by parasites. So it kind of preserves the ranges of the southern species. But I can't really speak to birds on that.
Yeah, the reality is we don't really know the full answer to that. So I think there are things, I think both things are true. There are times that, there's no way it's benefiting the bird. And there are situations where, just like Abigail talked about, we could imagine benefits. And I think, this is one of the things about this whole California condor thing. Matthew didn't give you all the fine details. The fine details were that literally the veterinarians we thought caused extinction of this Culticephalum californici, which is kind of funny, right? It caused an extinction event while trying to save another species. This is called co-extinction. And there are a lot of fancy publications in journals like Science about co-extinction and how we actually need to preserve parasites. And one of the arguments is that, you have a species like Culticephalum californici. It lives on that host. It lives in this environment with other parasites on it. What happens when you pull it out of that environment? What happens to the other parasites that are there? Is it controlling any of them? Like sometimes parasites eat each other. Sometimes they compete with each other. So if you pull one player out, then other ones can go crazy. And maybe that's why this introduced louse on Abigail's birds from chickens is going crazy. Maybe something is happening with the other louse on there. It's maybe affected by agriculture and the chicken lice aren't. It's a pretty interesting situation. So, in that case, then, the louse that is from that host, is potentially sort of helping to compete against these other invaders, maybe.
You kind of fingerprint birds based on these?
You mean based on lice?
Yeah.
If you give us a louse, we can often tell you what the bird is, yeah, or vice versa.
Okay. If I gave you two different louses, whatever the plural is, could you say for certain that they'd be sufficiently different in terms of did they evolve quick enough for something where you could actually see differences in the lice on there, or would you kind of just say, oh, they are?
I guess I'm not understanding.
I think I might get
Yeah.
what you're saying it. The morphology can be so specialized to a specific host and even to a specific body part of that host.
Yeah.
Like someone like me who's not an expert, based on the shape of it, I can tell you that thing definitely lives on a wing. Definitely is a generalist. Maybe it lives on the head. But I think with the slide with the tinamou, all of those lice have such crazy different body structures, I think, because of the diversity of feather types on tinamou.
That's one of the hypotheses.
Yeah.
Diversity of feather type.
So you can, even for someone who's not at expert level, I can already make some assumptions about what I'm looking at.
Tell me more about that. What do you see on a louse that tells you that it lives on a wing, or on another one that it lives on a head?
So
What do you see?
if these are barbels, maybe not barbels. but
Yeah,
something
barbels,
with
Yep.
The feather lice,
Feather
yeah,
barbs, yeah.
like his falco first, they will slot into the spaces. And that's kind of their defense against the host.
Yeah, basically, they feed on the rump and the back, but when the host starts to preen, they run to the wing. So doves have lice that are long and thin like that. And this morphology is where basically it's evolved independently over and over and over again. because, basically natural selection on avoiding host preening is so strong, because if you can't fit between those feather barbs, you're gone. The host preens you off.
And what about those on the head? What do they have?
Head lice, to me, I feel like they're the opposite bodies, that are chunky and round, and the bird, all they can really do is scratch. So they don't kind of have to be passed through such a fine filter as the bill.
So the bird can't preen as effectively on the head, so the lice.
And they're really good at grabbing on with their mouths. So they grab onto the feather barbs on the head with their mouth and just catch and hang on tight. And a little scratching claw is not gonna do much in comparison to a bill that has a little quick dip on it. So they have that other, that's their way of getting away. And
It's
some,
insidious.
yeah, it's pretty insidious. The other cool thing about the head lice on birds, if you look at a bird like a cotinga, so you take like, Amazonian umbrella bird, it has two lice that are in the same genus. One is called, Cotinga stotzi, and I forget what the other one is. One of those, lives on the body, it has a body morphology. One of those has the head morphology. Body morphology is black in color. The one that lives on the head is not black. And that's because, there's no selection for that, mimicry of the background, that background matching to evolve, because the bird can't see its head. It can't preen by visual sight of that parasite. It's just got a scratch. The other cool thing is those head lice on cotingas are generalists. They live on lots of different cotingas in the same place. The body louse that matches the color of the cotinga is only found on one species. So.
And the color matches for the.
And the color match. Well, we haven't looked at actually some of the white cotingas yet, but that's my prediction. Yeah, that the white cotingas.
Not the red ones.
Well, they have red patches, but they have a lot of melanin on them. So those have, like, purple throated cotinga has a black louse that lives on his body.
So they head for the block.
Yeah. And there's black underneath that iridescent red or iridescent purple.
At the base.
When you say generalist, what you just said is how I know the term generalist across multiple species, but I think correct me if I misunderstood. but
There are two
multiple-
generalist sorts of things here.
On the
Yeah.
body as well, you're calling
Yes.
generalist?
Yeah, so there's a generalist, it's called an ecomorph. So like a generalist ecomorph goes anywhere on the body. You might have there's a wing louse, a body louse, a head louse, and a generalist ecomorph. And then there's this thing on the left here in this picture of Matthews. That's actually, so all these specialist ecomorphs are in one suborder of lice. The one on the right is in that suborder. It's called Ischnosera. Try to spell that. And then the left one, those are called Emblicera. Emblicerans are these weird things that walk around, a lot on the skin underneath the feathers, but they'll come out on top too. And those don't really have ecomorphs. So we don't, they don't have that. The specialization is kind of different. They don't have this like body specialization on that.
A couple more questions on the chat. Greg has another question. Has any research been done on Andean condor parasites? And if so, how does that compare to California condor parasites?
I don't know any research done on Andean condors, like for the Falco-Ipirus thing that we're trying to solve. We tried to get lice from the skins of Andean condor, but we didn't get anything at all.
There are records of lice from those, but we haven't been able to get our hands on any specimens yet. Yeah.
Greg has another question. Is there research on the impact of insecticides on ectoparasites? For example, a pesticide that does not affect the bird host, but may affect its parasites.
Well, pyrethrums, that's what they were using when they killed these things. It's called 7 powder, or it's a powder, it's flea powder basically. And that's actually, that's made from chrysanthemums. So, in theory, it's biodegradable. It doesn't, that's what you would use on your dog, your pet, use it on birds. So it's in theory harmless to the birds. What's interesting is that some parasites have evolved resistance to these pyrethrums. So if you ever had lice as a kid, your parents probably put shampoo on your head that had pesticides in it. That shampoo doesn't work anymore. Basically, head lice have evolved resistance. The only way that you can get lice out of your kid's hair now is to get a louse comb and comb them out. Or you can use a fancy machine called an air Ali, which basically heats up their hair with hot air and blows hot air and basically dries the lice out. So basically you have to physically get rid of them or physically kill them. You cannot use pesticides anymore, unless you want to kill your kids. You can probably use some really nasty pesticides. But don't bother if your kids get lice.
How much research is being done on lice, on birds across the world?
Well, enough that every 4 years there's a congress. It's called the International Congress on Theraptera. And it's a group of people from all over the world that get together. It's about half people that study human head, body, and pubic lice, believe it or not. And then half people that study lice from wild animals like we do. And it's actually a fun group of people. And the meetings are usually somewhere exciting. You know, I went to Australia for a meeting, Argentina for a meeting. There are a lot of birders in the group too. So it's a fun group of people that like to get out birding. Well, actually, the last meeting that I went to was in Czech Republic, and I got to go birding in Hungary and Czech Republic on that trip. And the next one is in France. So there are enough people.
Where you can eat, we're along.
Yeah.
When's the next?
It is in not this summer, but next summer. So yeah, you guys can be thinking about giving a talk at that meeting. Perfect timing.
Any other questions in the room, on Zoom? Anybody on Zoom has questions, feel free to unmute and ask too. Give you 30 seconds.
I have a thing to ask about, but not anything to do with this. So remember.
Oh, the one thing I forgot to mention for our next program, Ken's talk, is that Head House Books is going to offer books for sale at the event, so you can buy your copy of the book from at the end of the talk and get it from the Ken. Gotta mention that earlier. Okay, no more questions on Zoom, no more questions in the room. Thank you, everybody.
Thank you. Does GDFC do a book auction?
Thanks to all the
All right, well,
speakers.
thanks
They haven't done
guys,
it.
very interesting. Thank you very much.
Yeah. Terrific first ornithological studies presentation in many years. Wonderful.
Greg, thanks for resurrecting the Orny studies. Appreciate it.
Well, you are so welcome, and thanks to everybody who basically said if I eliminated the committee, that was the end of me. So they were right. We need the ornithological in DVOC. So thanks to Jason and the students and everyone.
Thanks for having us.
Thank you.